The last group of receptors that we're going to look at is an incredibly large and diverse family of receptors. They are called G protein coupled receptors. Roughly speaking, these receptors can fall into three families. And I say roughly because I really mean roughly. There are different classification systems that are different receptors that fall into different classification systems. So on a very general level, we have a G protein coupled receptor, which will result in stimulation of a protein called adenylyl cyclase. We likewise have a group or a family of receptors that will actually inhibit the protein adenylyl cyclase. And then last, we have a protein that is actually going to stimulate a protein called PLC, phospholipase C. Those are the three main categories that we're going to be looking at in physiology. There's a fourth that is going to be involved in stimulating, let me find it really quickly. Oh yes, that one. Stimulating a family of molecules called the Rho GEFs. So okay, that's the number 4. So it stimulates a protein, or actually it's a family of protein referred to as the Rho proteins. Okay, and I'm going to acknowledge that that's there. And then I'm also going to tell you that there's subclassifications and so forth and so on. We're going to be focusing primarily on these three, okay, for our physiology class. And I'm going to actually even focus mostly on the stimulatory effects, okay? And we're going to look at these. But first, let's go ahead and define what a G protein coupled receptor actually is. First off, let's draw a cell membrane. Okay, so here's my cell membrane, and this is going to represent a receptor cell of any given type. We have my extracellular fluid out here, and we're going to have my intercellular fluid in here. Okay, so that's the orientation. The G protein itself, or the G coupled receptor, is what we call a it's a transmembrane protein, and it crosses the membrane 7 times. Now, we usually will have an extracellular region that binds the ligand, but not always. Okay, bear with me, not always. And then we're going to see this coil back and forth across the membrane. I lost track. I don't know if that's 7 or not. We're just going to add coils there. And then we're going to have a intracellular side that is going to bind a protein. So we have 2 binding sites, a ligand binding site and another binding site inside the cell that's going to bind its partner. This is actually what the coupled is all about. Okay? A receptor is this transmembrane portion, and so this right here is my receptor. Okay. Now on the inside, and I'm going to simplify this again. A lot of what I do, I'm overly simplifying it. There are situations where what I'm telling you is not actually technically true, but we're going to go with it anyway because many times it is true. Bound to the intracellular side, I'm going to have my G protein. Okay, so we'll draw it in an orange, and this is my G protein. And it's also called, let me make it a little bigger so I can have a little bit of more room to write. It's also called the alpha subunit. Okay, I don't have room to write unit, so we're just going to leave it at that. Now this G protein is a lipid anchored protein. You remember that? So I'm going to draw this little attachment here that is going to show that it's going to anchor into the fatty acid layer of the phospholipids. Additionally, this G protein is going to be bound to some other molecules, and these will be attached again to this alpha subunit, and these molecules in particular happen to be called beta and gamma. Okay, beta and gamma. And most often this is how everything's assembled together when the receptor is in its inactive state. Okay, inactive state. Now, let me actually erase some of this so we can use that there. In its inactive state, there's one more thing that is bound to the G protein. And then that is a chemical molecule called GDP. Guanine diphosphate, which is very closely related to adenine diphosphate or adenosine diphosphate, otherwise known as ADP. So in its inactive state, let me go ahead and jot this down. All of this together is this assembly part. Okay, and so this alpha and beta and Y is where we get the coupled term. The receptor is coupled to the alpha subunit, which is coupled to the beta and gamma. So in this state, it's inactive. Now, sometimes alpha is not actually hanging out with the receptor. That's the exception to the rule. Sometimes it's just close by. Okay? But we'll go ahead and keep going with this idea of it being attached to the receptor because that's a typical arrangement. Now along comes a ligand that would be released into the solution. And when that ligand is released into the solution in abundance, we're going to see a change. And the first step, so this is our inactive step, we're going to look at my cell membrane again. And let's draw my protein again. And it's going to cross my membrane. Like so. And when my ligand binds it, so there's my ligand. When that occurs, what happens is a change in my receptor. It's it's a chain reaction of events, and you can see I actually drew it subtly differently. Part of that was accidental, but part of that was also to kind of illustrate a point where we can see that the shape of the receptor changed just a little bit. And when the shape of the receptor changes, that has a snowball effect, if you will, and it's going to cause the alpha to also change shape just a little. Okay, so my alpha G protein changes shape a little tiny bit. And when it changes shape, what it does is it releases that GDP, and the GDP leaves. And in its place comes a high energy molecule, GTP. Let me give myself a little bit more room there. And GTP binds to that alpha protein. Let's go ahead and put beta and gamma in here. Okay. Now, what's interesting about that is when GTP binds to my alpha protein, the next step that we have, we'll go on to the next page. So this would be this is inactive, this is step 1, where GTP replaces the GDP. And then if we move on to see what else happens, what we see happen is when GTP binds alpha, alpha is going to let go of the receptor. My alpha G protein bound to GTP is no longer compatible with that binding. It totally changes the affinity. You remember molecular interactions talking about that affinity. Not only does it change the affinity of the alpha protein for the receptor, it also changes the affinity of the alpha protein for beta and gamma. And beta and gamma tend to leave oopsies, what did I do to this poor thing? They do not turn upside down. I'm not going to worry about fixing them. They leave. Okay? And they're going to go and do some really important functions. But because I'm trying to simplify this and minimize how much you need to learn, we're really not going to focus on beta and gamma. It does do some really cool stuff. But what I want you to do is follow alpha. And so step 3 is for this all to disassemble. Now alpha is going to begin to travel. And so if we look at what happens as it travels along the plasma membrane, it'll get farther away from that G protein coupled receptor, or from that receptor with its ligand bound, and alpha will eventually leave and travel far enough that it's going to bind to a large protein. And at this point, the identity of the protein we're going to keep very generic. Okay, so let's make it look like it's got a spot to bind alpha, like so. And alpha will come along, and it's going to slip right into that docking site. And when alpha is bound to this big green blob worth of a protein with its GTP, what that is going to do is activate this protein here. And when that occurs, this protein is going to begin to produce a lot of second messenger. Okay, and for now, again, I'm staying generic for a reason. I'll explain that we're going to look more closely at these, but this is going to produce second messenger. Which will then go on to activate other proteins. Now, what we then have is we're going to see a change here. And so if we go back up here to this would be step 4. The alpha protein, not only does it bind to all these different molecules, it binds GTP, it can bind the receptor when it's not activated, it can bind beta and gamma when it's not activated, and it can bind this other protein when it is activated. The thing with alpha, though, is alpha is an enzyme too. So it has multiple functions here. And the enzyme happens to be what we call a GTPase. So, in my next situation, with my alpha and my GTPase properties, is this. My alpha is going to change this molecule. It's going to hydrolyze the molecule. It's going to break it up. And in breaking it up, it's going to produce GDP. Okay? And so that would essentially be step 5. Okay, let's get rid of this part. And I want to move step 5. Oh, fine. I'll just do it this way. And then I want to move step 5 over. So we're going to go in the opposite direction. Once GDP is now bound to the alpha, what we're going to actually see happen is that alpha is now suddenly not active. And so it's going to leave that protein, kind of leave it all by its lonesome over here. And the moment it detaches to that protein, this is going to stop producing the second messenger. Meanwhile, that alpha subunit is going to go right back to the receptor and it's going to bind to that receptor once again. Okay. And beta and gamma are going to show right back up. Oops. And now we're back to our inactive state. Now, importantly, if the ligand is still present, let's say the ligand is still bound to that receptor, alpha becomes activated once more, binds GTP, and the process starts over again. But if the ligand is gone, then at that point, this alpha protein just kind of hangs out and it's done. And the signaling process is over. Now, this second messenger that I drew here is not going to linger forever in the cytosol. There are other enzymes. Let's make a Pac-Man enzyme. That will destroy any remaining second messenger. And so effectively, once everything goes back to inactive, the signal fades and we're done until more ligand shows up. Okay, so this is the basic process of G protein coupled receptor activation. By the way, the shorthand for that, because that's a mouthful, right? G protein receptor, G protein coupled receptors. Whoops, sorry. G protein coupled receptors. That's my abbreviation. GPCRs. Okay, GPCRs. So let's take a look at this. I mean, these are my scribbles, but I do have nice, neat, pretty pictures for you, okay, where you can actually see this assembled in a more beautiful format. I will add a little bit of information to this. As I mentioned, this is lipid anchoring. And so it's going to kind of hang out by attaching to that phospholipid bilayer, okay? And so you can see here that we've got several things that I'm pointing out. Here's my receptor, right? We look at this, my receptor. Let's go for a little lighter color so I don't overwhelm it. There's my receptor, okay? And you can see my alpha, right? So here's my alpha, okay? And you can see the GTP, and you can see the beta, and you can see the gamma. This is my inactive state. Now I want you to notice my ligands. Most of the time we get a ligand that binds out here in the extracellular fluid, and that will activate this process. But sometimes we actually have these ligands binding inside here. This region, remember, this whole region is hydrophobic, and so it can bind in the inside there. So most of the time the ligand is hydrophilic. But occasionally the ligand is hydrophobic. Which you'll remember the other word for that is lipophilic. Okay, so you need to be aware of that, and that's why I have the arrows shown you there, so that you understand that we are looking at two possible binding sites depending on the type of receptor and depending on the type of ligand, okay? Now, we can see then I've got my receptor circled. You can see my alpha and my beta and my gamma. And here I talk a little bit more about these, and I want you to know that the beta and the gamma are these second messengers, and they do cool stuff. But again, because of time constraints and as well as the fact that you guys aren't pharmacologists yet, I'm working on converting you, but you know, you're not pharmacologists yet, we're going to leave out some of the activity of the beta and the gamma and what they do. And we're going to focus primarily on what happens with my activation process. So here I break it down step by step. We have step 1, okay, where we've got the ligand binding the receptor, which is then going to cause the change. Sorry, I've got a few extra pages. I put these in here in case I feel like drawing. And sometimes I do it a little differently. So anyway, ligand binds the receptor. When the ligand binds the receptor, you can see here we get that exchange of the GTP and the GDP. When the GTP binds, that alpha disengages, beta and gamma are released. Beta and gamma go on to do their own little magical thing. And then here we see this generic, in this case it's a purple blob, but regardless, it's a protein that is activated by the alpha. And when that occurs, this protein would produce that second messenger. And usually it produces quite a bit of the second messenger. And so we say that this protein is an amplifier. It amplifies the signal and increases the magnitude of the signal. And this helps us so that we can get a big response to just a little bit of ligand. And so we'd have our second messenger. And it's going to be synthesized as long as that alpha and that GTP are bound to it. And the enzymatic activity of the alpha is relatively slow. So we're talking, this is going to go on for at least a few minutes. And then when we're done with that, we're going to see, notice that this GDP has now been hydrolyzed. So the GTP, to get to this state, this GTP was hydrolyzed. It was broken down by an enzymatic reaction. So GTP was broken into the GDP. And now we've got everything that's going to go ahead and assemble back together. Now, in a nutshell, every G protein coupled receptor works like that, with some variations depending on the type of receptor it is. But not all of them. I want you to look at this very interesting slide, not this one, I'm going to delete that one, but let's look at this very interesting slide here, and I want you to see what we can see from here. I don't expect you to memorize this slide, I don't. But we're going to see our families here. And I want you to look at this one here. It's hard to see what it says. So let's move in here and let's look at this. What this actually says is alpha I. And that I stands for inhibition. Okay? And what it does is it binds to a protein called adenylyl cyclase. Inhibits something called CAMP production. Cyclic AMP is what that stands for. It can also bind, interesting, it can also bind to ion channels and something called phosphodiesterases and phospholipases. But the I stands for the fact that it inhibits the adenylyl cyclase. Let's move in on here. I'm skipping Q for right now, and we're going to look at this one. This one, if we look at this, that alpha stands for alpha S, for stimulatory. And in this case, that alpha S also interacts with my adenylyl cyclase, but instead of inhibiting the production of cyclic AMP, which is a second messenger, it's going to increase it. So alpha I and alpha S oppose each other. Okay? And you can sometimes find them in the exact same cells. They're activated under different conditions, and what this allows is a fine-tuning of the control of the activation, the activity of the cell. We may have a situation where we're going to have some stimulation, but then alpha I would control that or dampen that and prevent it from being too much stimulation of adenylyl cyclase. And then this third one here is alpha Q. And sometimes linked with that one, so we've got this alpha Q, and sometimes that one is actually partnered with alpha 11. So a lot of times you'll see it written like that, alpha Q or alpha 11. And alpha Q activates a protein called phospholipase C beta, but I usually don't include the beta. I just usually write phospholipase C. And the activation of that causes these second messengers to either be created or released or activated. And then here's my fourth one that I usually don't spend any time on. I'm going to identify it for you, okay, and give you that name for it. It's the alpha 12. And sometimes you see this written as alpha 12-13. And you can see that that's going to activate these molecules called Rho. Rho GEF or just Rho. And those actually are growth, they're involved in growth and development of the cell, and so they're going to help generate these types of biological responses. Now, ultimately, all of these will result in gene transcription. Okay? Ultimately, they all do. Now, it's not always a direct path, but they all do this. And so they can cause these cells to respond in such a way that they produce more proteins. They proliferate, they differentiate, they develop, they survive, they grow larger, and when things malfunction, unfortunately, you can get cancer. Let me talk about angiogenesis really quickly, just to give you a definition of that. Angiogenesis and what I hope you do as you study and you learn and is you get used to looking things up. But this is to the development or growth of new blood vessels. If a tissue is growing larger, which look at this, we've got differentiation, development, hypertrophy, right? Cell division is involved in there. That's the proliferation part. If we're getting more tissue in a region, we need more blood supply. Unfortunately, if we are growing out of control because we have cancer, many cancer cells often tap into this process, and so we often get very large amount of blood flow to those tumors that are growing as well. So it's kind of interesting, and it's interesting from a pharmacological aspect in that often when we treat cancer, one of the things we do is we target angiogenesis to stop angiogenesis from happening so that we starve the tumor, so that the tumor doesn't get enough blood. Let's look at here, and this is an example. This is a family tree. I don't expect you to know much of anything about this except for look at this and be able to go like, oh, wow. These are the known, or at least known as when this was published, and I think I forgot to put, I didn't list my references, shame on me. This is from a review in the journal Nature, and you can see just how vast these are. I want you to see a couple of things that are interesting to me. I want you to notice this. Your olfactory receptors, the things that detect smell, are G protein coupled receptors. Many receptors that detect glutamate are G protein coupled receptors, and so forth. And so you can kind of see these. Well, this one's a fun one, rhodopsin. This actually allows you to detect light. So your light sensing receptors in your eyes, some of those are G protein coupled receptors. I think all of them are actually. Now let's look up here. I want you to look at these and notice what this is not a complete list, but look at what some of these ligands can be. We can have noradrenaline, which is also called norepinephrine, dopamine, 5-HT, histamine, acetylcholine, glutamine, calcium, GABA, We can have various lipids such as LPA, PAF, prostaglandins, leukotrienes. Now, a lot of these you haven't heard of yet, but we'll talk about these things. Some of them, not all of them. But, thrombin is one we're going to talk about. FSH, LH, TSH, endorphins. By the way, your endorphins are your natural opioids. They're not actually opioids, but opioid receptors bind to receptors that bind endorphins. I said that totally wrong. Opioid molecules, in some cases, bind to receptors that bind endorphins. And look here, this is just cool to me. Lights, odorants, pheromones, nucleotides, opiates, your cannabinoids. Okay, those are all part of this process. And so super, super diverse, which is really the purpose of this slide, is to just kind of show you that diversity. We're going to go on to the next video where I'm going to introduce to you some very specific, just two pathways. We're going to look at a very simplified pathway for alpha S and a very simplified pathway for alpha Q. Okay, those are the two things. Oh, before I move on, I almost forgot to mention that alpha I is also sometimes called alpha 0, O. So you'll see either nomenclature used in the scientific literature for that one. All right. So we'll talk about the specifics of these two pathways in our next video.